So I think we'll go ahead and get started. So good morning, everyone, or good afternoon, or whatever time it is where you're at. We're so glad that you could join us. I wish there was a perfect time for everybody. Sadly, it doesn't work that way. So this is the webinar entitled Pushing the Limits of Protein Evolvability, a webinar in honor of Professor Daniel Tawfik. And I'm really honored as the president of the Protein Society just to introduce, give you a pre-introduction to this session. I do want to plug the Protein Society. Registration and abstract submission is presently open for our upcoming annual symposium this summer in San Francisco, July 7th through 10th. We hope that you'll take advantage of the chance that I think we're all going to be able to get out and move around. It should be wonderful to see everyone in person this summer. Our next webinar will be quite soon, actually, in March 23rd. It will be organized. It's been organized by John Schlabag at Indiana University, and the topic is protein folding and misfolding on the ribosome. We have a wonderful panel assembled for that, so we hope you'll come back and join us for that. And I want to thank you for attending today. And before I turn this over to Lynn Kamerlin, I do want to say my own personal point on Dan Tawfik. So long ago, when I was a graduate student, I was really taken with the work of Jeremy Knowles at Harvard University. Dr. Knowles was an enzymologist, and he went on to be provost of Harvard and sadly passed away much too early. I was really taken with his work and with his thinking and style of doing science. And when he passed away, I reflected that it was sad that there was I didn't really have a Jeremy Knowles type person that I looked to as sort of inspiration. And so a few years later then, I read a couple of Daniel Tawfik's papers, and I very distinctly remember thinking, at last we have a new Jeremy Knowles to inspire us. And so with that, I'm going to turn this over to Lynn Kamerlin, who's going to introduce the speakers and organizers for this meeting.
Thank you so much, Chuck. So hi, everyone. My name is Lynn Kamerlin. I'm a computational biologist at Uppsala University and a friend and former collaborator of Dan's. So it was with great sadness that we as a community received the news of Dan's passing last year. Dan was an amazing scientist, but even more so, he was an amazing human being who impacted all of us profoundly, whether we were his former group members, his collaborators, his friends, or even if we only knew him through the amazing body of work that he left for us. And so many people I've talked to who I thought wouldn't really have any direct connection with him still have stories of all the different ways that Dan impacted them through his amazing science. So I really wish we were all here today holding this webinar for celebratory reasons, but I think at the same time that Dan would be really touched to see how many of you came here today to honor his memory. And we will do so through a combination of personal reflections as well as research talks from former members of the Tawfik Lab. So as an aside, before we continue, I just wanted to mention that we're currently working on a special issue of Protein Science in Danny's honor. And it's really nice to see how many of his friends, colleagues, and former group members have agreed to contribute to this issue. So we're wrapping up towards the submission deadline, and I expect this to be out in the next few months. And so we encourage you to keep an eye open for it for when it comes out in Protein Science. And so to start today's event off, it's my pleasure to introduce Dr. Lianet Noda from the Hebrew University of Jerusalem. She'll be moderating today's events, and actually she'll be introducing the speakers. And so Lianet is a former member of the Tawfik Lab who, like so many that Dan mentored, has gone on to a brilliant independent career. And it's our pleasure to have Lianet and all the speakers here today. And I thank all of you also for coming across multiple time zones to make this event possible. With no further ado, I pass to you, Lianet, and I'm very much looking forward to this webinar and to all your talks.
Thank you so much, Lynn, for the kind introduction, and thanks to the Protein Society, particularly Raluca, for organizing this webinar in Danny's memory and for making it all happen. I'm also very grateful for the opportunity to moderate it. It almost feels like a group meeting. Paola, Mikael, and Nobu, all of the speakers today, were postdocs in Danny's lab, and now they run successfully their own laboratories. I would recommend any of the students attending today joining this lab. any of these labs. I was actually very lucky to be lab mates with the three of them. And I always learned and continue to learn from them. So I'm pretty sure the talks today will be really spectacular and amazing. I see many or many known faces, or actually names in the attendance. And I think that to honor Danny as he deserves, the best would be to encourage you all to ask as many questions as possible to our speakers. Believe me, they will not be afraid. They are used to it. Our group meetings in the lab were always very, very vivid. Since this is a webinar, and then raising the hand and shouting your questions is actually not going to work as it used to work then. And also the microphones are muted. The way it would work here is that you actually need to write your question in the Q&A section that you will see below. And at the end of each talk, I will read some of them, so they will be answered live. And the rest, the speaker will do her or his best effort to answer them in the chat. And after the third talk finishes, or the last part of the seminar finishes, if questions remains to be answered, I've been told that, you know, we can of course consider staying a bit longer as wanted and needed. So I guess that's it for the technical part. And before starting with Paola today, I will open the microphone to Dr. Ita Gruic from the University of Zagreb. Ita was Danny's partners in the last years, and I'm very thankful she's willing today to come to share some memories with us. So you're next, Ita. Thank you.
Thank you, Lianet. Thank you very much. Is it okay, this presentation? You see it? Okay. It is very nice and at the same time very terribly sad to be here. It is nice because there are so many scientists and friends around the world that enjoyed and respected Dan's unique personality, his broad knowledge, his brilliant, as I used to mock him, gray cells, which helped him to make all these things in science. It is also sad, as I said, it's actually terribly sad because what we have now are only our memories. But how I like to think about it is that what we have learned from him about science, about friendship, about food, about how to enjoy life are inspiring for many of us. And this is his legacy, and I think the one he would be the most proud of. Sorry, it doesn't work, my presentation. Okay. Excuse me. Okay, so I would like to start with a picture showing Yitzhak Rabin, the late Israeli Prime Minister, laughing at a show that was actually sarcastic on his account. And that was the picture what Dan sent to me. And he was very much touched with this picture. And we discussed at length how it is really important to be able to laugh on your account and not to take yourselves too seriously. And I think Dan was really good at it. And this made him sincerely humble and approachable for many people, many students, scientists. But as much as he was not taking himself seriously, he was very much taking science seriously. And science was something really central to his life. It was sometimes even annoying to see how easily he was navigating through different scientific ideas from one to another scientific project, because it was enabled by his really deep understanding of principles in biology and chemistry and his really broad knowledge. He was very fast in thinking and writing, as many of you know, and he was very productive. Yet he was aware that science could be a cruel mistress, and we discuss it also very often. He sent me this quote from Jacques Monod stating that in science, self-satisfaction is death. And if we want to do if we want to work and do a good science, that agony of mind is something what we need to accept in our life, as Monod actually very beautifully said. And he was accepting it. He was really he was striving to do the best in science at the expense of his, at the expense of the peace of his mind and restlessness. His group was very important to him. This is a photo of his last group, and this is one lab retreat. He was engaged hours and hours in lab discussions, in discussions about science, in discussions how to, promote things in science in the lab. It reminds me of one anecdote. He told me when he was in States that when students approached him, when he was visiting some university in States, when students approached him and told him that he was following the idea which Dan gave to his boss, and the idea didn't work, and then he spent a couple of months working on it. And Dan smiled at him and told him, "Oh, you were lucky. Some have spent much more time working on something like that." So I just want to emphasize that he was never taking science as a safe path. He was always doing science and encourage other people around him to do science in a way that you ask important questions, which are very often very challenging and unsafe questions. He really liked to laugh and to take life with humor. And this is also a beautiful photo from lab gathering. And you can see his, I call it infectious smile, because you can see it on his face. It is radiating with, smile and happiness. He had a great sense of humor, sharp, a bit cynical, but never offense. Another anecdote, what was for me very cute, is that when he was writing a review, he acknowledged the Israeli rail company for long delays that enables the review to be written. And of course, when he sent this review, the editor didn't like it a lot. He said it could be a legal problem. So he eventually removed this acknowledgement. But this was his way, to deal with annoying situation. He tried to deal it with the humor. Talking about Dan and not talking about food is really difficult. So hummus was a religion, and this is something what he would and this is something what he would call the post-hummus smile. And he cooked really, besides hummus, he cooked really excellently. So very often this was something pretty simple, like this tomatoes or simple pasta. But believe me, it was really, really difficult to reproduce it at the same quality level. He also liked to decorate food. So he would say that the food should be joy for eyes as well as the stomach. So it was very often, it looked really, really nice when you got it on the plate. And it was really delicious to eat after that. He was a man of nature. He liked to walk, he liked to hike, he liked to climb. But he also had an eye for small hidden beauty in the nature. This is something like haiku photo, what he took at some of his walks or hikes. So some small, beautiful things, nothing grandiose. He was less man of sport. I think because of that, he was so proud of his climbing. He really liked climbing. He said that it gives him ability to detach his overworking brain, restless brain, that it can be detached only through this challenging activity like climbing, and it always refills his battery. He was very much into music. He was listening and know lots about the jazz, rock, classical music. He was reading a lot. From fiction to different non-fiction, scientific or politic or war non-fiction. This is his cat taking care of the book on his table. And he was also engaged a lot in politics, always thinking and talking about politics. Dan was very deep and thoughtful person, and yet in a way very simple. It's sometimes contradicting, but I would put it like he likes simple food, he likes simple flowers, but not simple thoughts. So he had many, many deep and very nice and important thoughts. He was full of excitement for science and life. You can feel the burst of his excitement very often. And to finish, I'm sure I can say in the name of many of us that he is and he will be missed dearly. And I'm really thankful and grateful to Lynn to organize this and for giving me the opportunity to share my thoughts about Dan and to represent him in this way here. Thank you very much.
I want to thank everyone for being here. We have more than 350 attendees from all around the globe, from New Zealand, Japan, States, Canada, Europe, all around. And it's very nice to see friends, colleagues, and very, very touching and exciting. So I will just present our next speaker, Dr. Nobuhiko Tokuriki, who did a PhD in, who has his own lab now in the University of British Columbia in Canada. Nobu did a PhD in protein sciences, and I guess biophysics and biochemistry mainly. And at Danny's lab, he did a fascinating work exploring experimentally and computationally very interesting questions related to protein stability and robustness to mutations. And of course, about protein evolvability, which is also at the heart of my own interest. I think it was at the heart of all of us in the lab. And he will be speaking more about today. So thank you, Nobu.
Hello, can you hear me?
Yes, now we can hear you.
Yeah, sorry. Can you see the screen okay?
Yes, the screen looks fine. Thank you.
Okay. Thank you very much for the introduction, Lianet. And also thank you very much for everyone coming in. And thank you, Dean and the Protein Science to organize this webinar. So I'm very honored to be part of this webinar. So it's great pleasure to represent what's Danny's legacy, as Mikael said. So this is kind of only picture I think I have with Danny. So it's kind of about 18 years ago. So actually, a few months after I joined Danny's lab, this is kind of really pleasant relationship between us. It's like really lots of talk and discussions about science and everything about life and cultures. So that's why our common interest is very deep and fundamental. It's simply just asking how do proteins evolve. So it's very ambient questions, but is there something I try to kind of find some key mechanism and trait that's we can capture, then it's hopefully turn into our ability to protein engineering and designs. So I want to also mention that why this journey
I think we have a problem with the Nobu.
No, you were fine.
We lost the audio.
Exactly. The last word I heard was this journey, and then suddenly the audio cuts.
is okay.
Yes, now
Yeah.
it works again.
Yes.
All right. Hopefully that's okay. So let's start by this journey between in this very basic question is coming from the many discussion around the world, and as I'm very proud of being a part of it. So this is one of the book I want to introduce here is the Anders Wagner's book. This is a very inspiring book. And me and Danny took a lot of idea from the book and then lots of discussions starting from those publication from different peoples. So the concept here I want to discuss also the theme of this webinar is evolvability. It's a very ambiguous concept. It's what it means is kind of the organism or the proteins or the genes, it's able to evolve, right? So that's kind of a trait is, or evolvable trait is selectable trait or even existing in biological system. This is a big question. It's still answer is as unknown, but there are certain traits that we can discuss in a biophysical and biochemical way that's probably related to the protein evolvability. We will be able to do some good protein engineering. So the question is, what are the mechanisms for the protein evolvability here? So you can see this climbing the fitness landscape, and if you want to climb to the peak, you don't want to stuck in sort of like a local maximum. So we want to avoid it, and we want to get into the peak. So there's the two aspects I want to talk in today. So the one aspect is how much protein can tolerate mutations. So that's what essentially we need to have the mutation to do the evolution. So it's kind of protein robustness and how much mutation can be accumulated. But another side is how protein can alter function quickly. So it's like whatever the mutation accumulates, it doesn't matter if it doesn't change the function when you looking for certain innovation or functional change. So this accessibility to the adaptive mutation is very key. And that's kind of connected with those two are connected, but there's a different properties we can talk because this is kind of the simple diagram of the protein sort of trait or functions of fitness. So when we're looking at an organism, it's sort of the flux of the chemical reactions, but its mutations are actually
Nobu, apologies, but we lost you again. We lost the audio again.
Oh, sorry.
Oh, you're
So
back.
maybe,
You're back.
no, that's doesn't look good. So I will cut the. Let me change the. Maybe sorry, stop sharing. Then. Do you still can hear me?
Yes, we can
Yes,
hear
we
you.
can
Okay,
hear you.
so
Yes,
yeah,
now we can hear.
switch to the computer microphone. So hopefully that's okay. Sorry about it. So what I was saying is the mutation affected of two basic properties of the proteins. One is the KKT over KM, the protein functions, and the KD and the protein binders, and also affect the protein stability and the functional expression. So there's this kind of mutation is two sides of the coins. It's affect obviously the protein properties. In the end, what we are looking for the flux or the activity in the cell or the organism duplication rate. So there's two factors. It's actually affect the two different evolvability factors. And then what we have to see is both effect is how it works. And just to note that my general concept, the mutation affect more to the stability. Almost any mutation along the proteins can affect the stabilities, but the functional effect is relatively small, and the mutation change the function generally around the active site or some kind of allosteric regulation. The fluctuation of the mutation affecting functions are relatively small. So I want to talk about the two aspects of the protein evolvabilities. One is mutation tolerance, another one is accessibility to adaptive mutations. The first talking about mutational tolerance. So this concept was really started from Dan Hartog, Dan Bainwich in the very beautiful review in the Nature Review Genetics in 2005. And then also the J.C. Blumen and Francis Arnold and our Danny's group done this massive job in establishing the concept. So the concept is very simple. There are certain thresholds that exist in the protein folding. So it's sort of the protein biophysics. The mutation accumulate and stability always fluctuates. Some mutations go lower the stability, some goes up and then just deviate. So this, but when the protein stability close the threshold, obviously we see protein unfolding, aggregations and degradations. So it's these mutations not going to be accumulated during the evolutions. Maybe some too much stabilizing, it's not great, losing the dynamics activity regulation, but this part is relatively ambiguous, but lower part is the very, very definite biophysics in the proteins. So lowering it is definitely the clearly easy to understand, and that's also the very simple problem because most of the mutations are destabilizing. So this kind of distribution of the mutation effect in all possible proteins, so this the black ones is computed by the Foldex. So you can see that most of the mutations has the plus delta G effect, which means a destabilizing. So the most of the case, those highly deleterious mutations will be purged out during evolutions. But when you look at the new mutation function, this is kind of hundreds of mutations people identify in directed evolution. And those look like it's almost all possible mutations. That means that a highly destabilizing mutation exists in those new function mutations, somehow accumulating directed evolutions. But obviously there are some stabilizing mutation as well, so it's kind of win-win situations, but overall tendency goes toward destabilizing. So the problem's always coming into that kind of closing the threshold. So simply, or we can think about how we overcome this. We can obviously stabilize proteins. There's a very common now strategy for the protein engineering. So you can start from the highly stable proteins. And also what I studied was that you can use certain chaperone or environment change to buffer or change alter this threshold level. And then maybe we can accommodate these destabilizing and functional changing mutations. But the problem is this strategy is probably very temporal effect, right? Once you got close to the threshold, even starting from stable proteins, you will have the same problem. So it's kind of, we have to keep solving the problem, doing something or getting stabilizing also compensatory mutations to cycling this process. Probably this is what happened in nature in the long term, but also we can do in the protein engineering or directed evolution scheme. So I don't have enough time to show all these details. This is kind of one example. I did it in directed evolution with Danny. So there's kind of 18 rounds of evolution. It looks like a very smooth evolution, but actually I manipulate quite a bit overexpression of chaperones and getting a stabilizing mutation. That's only the way we get to a very massive improvement, more than 10,000-fold improvement activities, and then we can get this like a nice transition between the native enzyme to the new substrate adapting. You can see the active site really adapted to the new substrate. So this is kind of the manipulating is a very important process in protein engineering. So this is kind of this side mutational tolerance. So if you manipulate it, you can probably elongate your evolutions. You can find more adaptive mutations here. Then when we've done this work, I thought, okay, this kind of to solve the big mystery, and I can do the lot of directed evolution to see big transition. But it wasn't really easy to do it, and quickly I realized it's many directed evolution is still not working, even starting from very stable proteins and the manipulating stability really still doesn't affect. So there's some kind of other problem and accessing adaptive mutations or increasing the function in fundamental level. So I'm going to talk about this a little bit in the last minutes. So one way to understand and seek this kind of problem, simply we have to do many directed evolution experiments. So there's, I just put the 4 excellent papers, that's inspiring papers done this parallel directed evolutions. It's coming from Davis's group and Wagner and Arnold and Liu's group, also the Joe Thornton and Dickinson's group. So what people can do is simply do parallel directed evolutions. Let's say, well, there's a kind of genetic diversity here, and there's a native functions represented in orange. So if you want to get the green colors, okay, why don't we start from a different starting point? So we are testing a genetic starting point and what kind of adaptive mutation exists in this system. So if you do that, and maybe you see certain positions or genotypes can access to the nice evolutions, but maybe the other position is not working. So that's people started to test it, and probably this one of the Davis's papers, one of the first one, and started to do it in a protein level, but also the many people started to engage in these questions. So we done similar experiments. It's a little bit minimum scales because our system is not really high throughput. Some of the recent work is a very elegant high throughput system. But what we did in here is, this is the directed evolution I showed earlier. So take this enzyme, actually mutate the first mutation we observe in the different amino acids, and it's the continued evolution. So that's by changing the single first step of the mutation, what's the effect of the outcome? So it's interesting enough, evolution actually stop in the middle plateau. So it's kind of, this is more than 10,000-fold improvement. If you start from, if you mutate the serine in these key positions and just stop, literally stop in 100-fold improvement. As I can say, that the stability wasn't really a problem, and it's just we can't find more mutations to evolve from this variants. Similar experiment we did with a different enzyme system. This case is the beta-lactamase. We took the 4 different homologs showing this the PMH activity in a different level. In the normal sense, if you're a PI, you would ask the student, okay, why don't you take this green one to start, because it's already the best variants in this system. But the Florian Bayer in my lab is just insisted, oh, let's try the different variants. And it's interesting enough, the lower activity variants actually quickly exceeded the best variants. And then actually evolutions went farther, way higher than this, that the best variants have been to. So there's something different between starting points. Well, this case is actually the two enzymes differ 70% amino acids, so it's a massively different enzyme, but it's exactly the same fold. So it seems to be accessibility to adaptive mutations are quite different in different enzymes, and it's obviously the question: what's the molecular mechanism underlying those phenomena? The simple answer is mutation epistasis. So probably many of you knows this concept. The mutation epistasis, the effect of the mutation you see is different when you have the other mutation or the other sequence to start. So most of the simple case, one mutation A, one mutation B. When you put the 2 mutations together, effect of the mutation is exactly additive, or the multiplication of the results. A certain case, 2 mutation makes better than the additive, or a certain case, the worse than additive. The worst case, the 2 positive mutation combination end up in a negative result. So this is like kind of 2 mutational presentation, but if you can imagine 300 amino acids interacting with each other, then that makes a very complicated situations. And if you see 70% different starting point, you can imagine these things happens a lot. So directed evolution in our system is telling us that epistasis exists a lot, because when we did this 2 evolution, you can see mutation accumulated, or the adaptive mutation accumulated each trajectory, it's completely different in each other. So these two trajectories, for example, only one position, only one amino acid shared exactly same mutations. 3 other positions same mutated in 2 trajectories, but it went to the different amino acid. And other mutations are actually completely different positions. Same things happen in these 2 trajectories we did in this beta-lactamase. You can see 2 positions are mutated in 2 trajectories, but other mutations completely different positions and different mutations. So somehow when you start at a different starting point, even a single point mutation in this case, you can actually completely alter which mutations are adaptive and afterward. So what's the molecular mechanism? And obviously it's causing epistasis and evolvability. That's kind of the bigger question we are tackling in the lab. I had to say it's very, very complicated, and I'm not smart enough to understand this phenomena, but we try to get some answers. So this case, for example, what we have to do is obviously see what's the change between wild to the evolved variant. So this kind of successful variants on the beta-lactamase evolution. So you can see the wild type has the kind of this pocket is kind of clashing with the substrate. This is actually the substrate it's the complex with this evolved variant. So evolved variant has a very nice pocket accommodating this the phenol part of the substrate. And then you can see the substrate is nicely located in the active site. When you look at the wild type, this is not going to happen because there's a big the clash in here. So it's most likely substrates have to sit in a different way than this evolved variant. So the evolution is most likely solved making the pockets and sitting, like accommodating substrate inside. So the evolution improved actually 20,000 for the kCal. You can see the actual expression in the soup it suffers a lot. So this is a classical example of the functional stability trade-off, because they have a nice solution for the adapting the substrate, but the stability suffers a lot. Other one, unsuccessful one, is very weird things happens. The activity only increases 124. Actually, the expression is getting better. So it's the stability is not the problem for this enzyme. But then when we solve the crystal structure, actually we see the swapped dimer here. It's even doesn't look like active enzyme. It's like one of the metal is gone, for example, here. So very weird things happen, and it's on this unsuccessful starting point. So this way, BIM actually exhibits the initially high activities. Stability is not really a problem, but somehow NDM makes the very nice accommodation of the substrate. So why this happened is very, very subtle. So we ideally can't answer this question. Actually, the mutation making the pocket is a very simple one. Actually, there's a tryptophan in here, and it's remutated to glycine. That's actually improved 100-fold activity level. So it's making a nice pocket here. But this is actually the tryptophan in the BIM as well here, and it's slightly different orientations, but mutating actually this BIM tryptophan into glycine actually doesn't do anything in this evolution. It's really the devil in the detail. That's like similar to what Mikael showed in previous talk. We really probably have to understand how enzyme interact with the substrate and what's really the problem this promiscuous substrate sitting in the active site. And it seems to be successful evolution example solving that problem. And unsuccessful evolution cases, maybe the problem is much more complicated and a simple mutation not giving the answer for this. So it's like that's just summarizing my talk is we really have to find both nice part of this evolvability has to be obviously stable enough protein to tolerate mutations, but we have to find certain scaffold that has more accessible to the adaptive mutations. So I just wanted to acknowledge all the members in my lab. Also, this Danny's really inspiring. There's a lot of discussions to these so many ideas. We have so many unsolved questions. So I really encourage the young people to come into the field and then work together with us. And thank you for listening.
Thank you, Nobu. That was a very, very nice talk. I have questions here for you. The first one that popped in is from Adrian Hugelmatter. He says, hey, Nobu, what happens if you insert the serine mutation in the more evolved pathway on the arch, the ones that started with arginine?
Yeah, so if you mutate, initially the serine mutation is almost neutral, so it's almost equivalent to the arginine. If you mutate arginine to the serine in the last evolved variants, enzyme essentially lost all the activity gained during the evolution. So that's arginine definitely needs to be there.
I have the next question from Peter Birning. It says, I'm curious about what methods you are using for directed evolution. Is it DNA shuffling or error-prone PCR or something else? Also, it would be interesting to see the changes in protein thermal stability over multiple rounds of directed evolution.
So this example we showed, we used, like you said, like error-prone PCLs and DNA shuffling. So the very simple mutational inputs. The other people doing like more continuous evolution is probably the more like simple, like the error-prone PCR type mutations. But that's what we do. We follow some of the protein thermal stability. So there's one example I showed. This is really up and down during the evolution. So once functional mutation accumulating, actually stability a little bit goes down, the more expressions goes down, and we actually need to put the stability mutation to compensate. So this cycles definitely needs to work in certain example.
The next question from Aravinda Munansigye. Do you see any relation in preferred expression levels with changing fitness of the protein?
Some of the case expression going up as directed evolution goes. So that's contributing to changing sort of protein fitness. So that happens. I believe that happens when we kind of run out of function changing mutations, and then protein evolution will go up. And that's kind of maximum evolution can do to this gene. That's our hypothesis, yeah.
So I will take one more question and then maybe the rest, if you have time in the chat, Nobu.
Yes. Yeah.
The last one I will read is from Liam Longo. It says, is folding cooperativity and well-defined structures a major enforcer of a strong epistasis? Or will ancient flexible proteins be less epistatic?
That's a difficult question. So most of the protein we work on really not cooperative folding proteins. Most of them actually very aggregate prone proteins. So it's obviously a cooperative folding, but this folding seems to be very complicated. And cofactor is a very important factors for the activity. I wouldn't say this simple dogma. I don't like the simple dogma like flexibility, changing epistasis, or the evolvability. Probably much more complicated things happen. So epistatic, it's very not really common when you look at the two mutations in a protein. If you pick the two mutations randomly from the proteins, they are most likely not epistatic. So we see massive epistasis when we're looking at the inactive zones. So the flexibility in the primordial proteins can be either way. I think that really depends on the protein fold and the gene itself.
Actually, maybe I do have a time for another question. It says, it's from Daniel Weisman. It says, even conditioning on the first mutation, different runs will fix different second, third, etc. So how much spread is there among these different trajectories? How does it compare to the difference among the means of trajectories starting with different first mutations?
Yeah, so we haven't got all epistasis data, but it's look like that's where the first mutation opening up the second mutation and third mutation, fourth mutations. Not always clear order, but it's early mutation definitely promoting other mutations. We have some epistasis data, but also structurally very makes sense. Actually, the first mutations comes in and second, third mutation seems to be stabilizing the position of this first mutation. So that kind of first mutation is actually interacting with the substrate and partially open up the solutions, and another mutation kind of helping to kind of reinforce and stabilizing the active site configuration. So that's one case we see it. Not all the case we are seeing epistasis. Some cases are actually non-epistatic as well. We try to get a bit more systematic way in the big community libraries, and hopefully we can tell in sometime in the future.
Great. So I will stop asking questions to Nobu now live.
Yeah,
and
thanks.
Also to the rest of the people that still want to participate, feel free to continue asking questions in the chat, in the Q&A part, and Nobu, Mikael, and Paola will be, very happy to receive those questions. Next, we will have Stephen with us that will tell us, Stephen Fry from the Johns Hopkins, and he will tell us about some experiences he had with Danny recently before he passed. So thank you, Stephen, all yours.
Yeah, thank you, Lianet, for the introduction. And I also, of course, want to thank all the speakers who gave amazing, beautiful, diverse talks that I really think illustrate the range of interests and talents that Danny had. What I wanted to do actually with this group is just share a little story, hopefully don't want to take more than a few minutes, but like many of you, when I first heard about Danny's untimely passing, I sort of reverted to the sort of normal, human instinct of trying to, recall what my last interaction with him was and try to find some meaning in it. And in my somewhat unusual case, that final interaction actually occurred after he had passed away. It happened because Dan Tawfik was a reviewer on a manuscript that we had submitted to JAX. And in my case, Danny was the dutiful reviewer number 1, who submitted a very extensive and very useful review on time. And some slower reviewer 3 took many more weeks to get it completed, such that when the editor of JAX sent us the review, it had the illusion that he was providing scientific advice from the other side, so to speak. So it was one of the more unusual and uncanny moments in my career so far. And I thought I just wanted to take a moment to share with the group, what meaning I extracted from that experience. And so the question I basically want to pose to you is that if you knew a review would be your last, would you write it differently? Would you try to inspire and teach a younger scientist like Danny did? Would you stand up openly by your critique and put your name at the bottom of the review like Danny did? Would you try to tear someone down, or would you try to build them up, as Danny clearly did in this situation? And of course, Danny had no way of knowing that this would be his final review. The fact that he put so much time and thought into it goes to show how thoughtful and caring he was about every scientific interaction that he had. So I think if there were 2 lessons that I could extract from this sort of unusual circumstance, the first one would maybe be a sort of a tune on a classic one review not how you would not have yourself reviewed. I think that is definitely apparent. But I think also more broadly, it sort of shows me that your scientific legacy is not just what you've published, that all of the interactions that you've had with other scientists, all of your personal communications, even anonymous or not so anonymous reviews have an enormous impact on the future of scientific posterity. And I think something about Danny's review from the other side kind of made that feel very real to me. On a slightly more humorous side, in Danny's review, he took exception with the fact that I had one sentence in the manuscript that said that these are, this is a certain fraction of the number of proteins that are believed to be expressed in E. coli. And he said, don't use the word believe. That's not, the right word to use in that context. And he points out that I assume that this number derives from scientific work, not a prayer book. So I think it's worth pointing out in this context that, like many secular Israelis, Danny expressed Jewish cultural mores and was actually relatively familiar with sort of key Jewish teachings, even though he did not adhere to its many restrictions. And so I just wanted to end by sort of sharing with you one lesson that Danny shared with me when he was still alive, and something that we actually spoke about when he invited me kindly to his home for dinner and actually shared some of the amazing foods that Itza had some beautiful pictures of. I just wanted to share this with the group. So this is a very well-known line in the rabbinic literature. And here it is in the Hebrew. Below, I have it translated in English. And it goes like this: every dispute that is for the sake of heaven will in the end endure, but one that is not for the sake of heaven will not endure. So Danny and I spoke about this. It's interpreted to mean many different things in the Jewish context, but we were really interested, what does this have to say in the scientific context? And the interpretation that Danny gave to me is that what this sort of is saying is that just because you win an argument, it doesn't mean that you've won. Instead, if an argument is to serve a higher purpose, then everyone wins. And so I think this science, especially Danny's kind, is a dialectic. It thrives on disagreement and arguments. And arguments can sometimes be bitter. They can sometimes be difficult. But we need to have those arguments. That's how, of course, science advances. And so I think if you the key lesson that Danny leaves me with is that if you're ever about to engage in a scientific argument with someone, ask yourself, is this an argument for the sake of a greater purpose? Are you both trying to understand a fundamental mystery of nature? Are you respectfully trying to question someone's assumptions? Or is the argument not for a greater purpose? Are you arguing over who gets credit? Are you arguing over whose method is better? And in so doing, that's not an argument that will endure. It's not one that will leave a legacy to the scientific process. So that was just something that I wanted to share, just because I know that many scientists don't come from traditional Jewish backgrounds. And so perhaps this is a side of Danny that he didn't always share with everyone, but he did share with me, and I wanted that to be something that more of you could experience.
Thank you so much, Stephen. That was really touching, amazing. Thank you to all the speakers for the great talks, to the Protein Society and to Lynn for allowing this webinar in the memory of Danny to happen. How can I say? I think we all miss him very, very much. I still mentally consulting with him all the time. And it's just sad that I won't get a real answer anymore. But I hope that through his former group members that are now spread all around the world, opening labs, and his former colleagues and friends who are most of them scientists, we can keep his vision. And I just want to remind you all that part of it will come out of in this special issue of the Protein Society, where mainly work related to what had to be with Danny's open questions in the field are going to be, or related to his interest in science, would be put all together. And if you want to comment something more about, Danny's, or share some experience, the Q&A is open now if you wish. And I don't think that will, well, I don't know, it doesn't seem that would be the case, but I just leave stage to Lynn. Thank you so much, Lynn, for this, and all of you for attending. It was very spectacular to see how many participants we had from so many different countries. Thank you.
Thank you so much. I'm going to start on behalf of the Protein Society by thanking Lianet, all the speakers today, and every one of you who attended. So I've been monitoring the participants, and at our peak we were 360, and people logged in, at least the people who I know where you are, people logged in from as far away as New Zealand at 4 in the morning, all the way through to Vancouver and possibly beyond, and that was great. The other thing that was really nice to see in the audience was so many familiar names of friends, of colleagues, of collaborators, of former group members, scientific children and grandchildren. And it was just really, really touching to see such a wonderful group of people come together to honor Danny's memory. And I think if he were here to see you, he would be so touched to see all of you here. So thank you very much for coming. As a reminder, we're also doing a special issue of Protein Science, which should come out in the coming months. So keep an eye open for that. I hope to see you all in person in San Francisco for the annual Symposium of the Protein Society. It's a great lineup of speakers. San Francisco is an amazing city. If you're there, please come and say hello. And otherwise, have a wonderful day. And thank you again for attending.